Welcome to the deep dive. We're plunging into some fascinating territory today. We're zooming right in on, well, one of life's biggest mysteries, really how it all begins.
Those very first moments, yeah, the creation of a new organism at the cellular level, it's pretty mind-blowing when you get down to it.
It really is a dance of incredible precision.
So for this deep dive, we're exploring gametogenesis and fertilization. Basically, our mission, for you listening, is to unpack how those building blocks, the sperm and the egg, are actually made.
Right, the cellular choreography, as you put it. And then what happens when they meet? That cascade of events leading to the very first cell of a new being.
Exactly. We want to give you a clear picture of these, complex biological steps, but without getting totally lost in jargon.
And we're drawing our insights today from some detailed embryology notes. So it's like having a front row seat, looking through the microscope.
A microscopic view of life starting. Okay, let's dive in then. Before anything else, before fertilization can happen, these specialized cells, the gametes, sperm, and eggs, they have to be created. That process is gametogenesis. And what's really striking is that the basic genetic shuffling part, meiosis, which halves the chromosome number, it's similar for both sperm and egg development.
Similar process,
But the end results, vastly different cells. Completely different structure, function, even you could say different biological strategies.
Okay, so let's start with the sperm strategy then. Spermatogenesis, the journey of sperm formation. Where does this happen?
It happens in the testes, specifically within tiny coiled tubes called the seminiferous tubules.
Right, and inside these tubules.
You've got two main players. The germ cells, they're the ones that actually go through meiosis, the cell divisions. And then you have the Sertoli cells.
Sertoli cells. So, okay, what's their role?
Think of them as the crucial support system. They nourish the developing sperm, provide specific growth factors. Basically, they manage the whole environment. They're essential.
Got it. And these germ cells, they don't just appear there, right? I read something interesting about that.
No, that's right. The primordial germ cells, the very earliest precursors, they actually migrate to the gonad, the test in this case, from other parts of the developing embryo. It's quite a journey.
Wow. Okay, so they arrive, and then what?
Then they become spermatogonia. These cells hang out near the outer edge of the tubules, and they start dividing, just regular cell division, to increase their numbers.
Okay, multiplying.
Yeah, and then some of them grow larger, becoming primary spermatocytes. These are the ones committed to meiosis.
And before meiosis starts, there's that crucial prep step, right? The DNA.
Exactly. DNA duplication. Each chromosome makes a copy of itself. So the primary spermatocyte has double the normal amount of DNA before it even starts the first meiotic division. It's loading up.
Loading up for the big genetic shuffle. Meiosis 11.
Precisely. And during meiosis A, something really key happens. The duplicated chromosomes find their homologous pairs, the one from mom pairs with the one from dad, and they line up together really closely. This forms a structure called a tetrad.
4 strands all bundled up. And this close contact allows for
crossing over. This is, fundamentally important. It's where the paired chromosomes literally swap segments.
they break and rejoin.
Yes. Corresponding segments break off and reattach to the other chromosome. It's a physical exchange of genetic material between the maternal and paternal chromosomes.
And that's the source of genetic recombination, mixing things up.
That's exactly it. It shuffles the genetic deck, creating new combinations of alleles on the same chromosome. It's why siblings, apart from identical twins, are genetically unique. It ensures diversity. 2 of the resulting chromatids will be recombinant, carrying these new mixes.
Okay, so after that shuffling in meiosis the primary spermatocyte divides.
Right. It divides into 2 smaller cells, the secondary spermatocytes.
And they immediately go into another division.
Yep, meiosis the second. Each secondary spermatocyte divides again, resulting in 4 even smaller cells called spermatids. Now, these spermatids are still roundish, not quite sperm-like yet.
So there's another step.
Yes, and it's distinct. It's called spermiogenesis. This isn't cell division, it's a transformation. The round spermatid dramatically changes shape, sheds excess cytoplasm, grows a tail.
Ah, the streamlining process.
Exactly. It remodels itself into the familiar modal sperm cell. It's crucial to separate spermatogenesis, the whole process, from germ cell to spermatid, from spermiogenesis, which is just that final differentiation stage.
And the overall strategy here is clearly about numbers, right?
Absolutely. In most vertebrate males, these spermatogonia are like a factory, constantly dividing and churning out huge quantities of sperm throughout adult life. It's a numbers game, maximizing the chances that at least one will reach the egg. Mobility and quantity.
Okay, so millions of tiny mobile sperm. Now let's contrast that. Oogogenesis creating the egg. How does that differ?
It's almost the polar opposite in strategy. Oogogenesis isn't about quantity. It's all about quality and resource accumulation.
Same starting point, though. Primordial germ cells migrating.
Yes, the origin is similar. Primordial germ cells migrate to the gonad, the ovary, this time and become an oogonia. But then things take a very different turn.
How so?
The growth phase. Some oogonia grow, really grow, becoming primary oocytes. And the scale of this growth can be just astounding.
Astounding how. Give us an example.
Okay, get this. In some animals, like birds or reptiles with yolky eggs, the volume of the primary oocyte can increase by up to 100,000 times.
Wait, 100,000 times? That's
Yeah.
enormous. Why? What's the point of getting that huge?
It's all about packing supplies. That massive growth accumulates vast stores of RNA, proteins, lipids, yolk, and everything the embryo will need to develop after fertilization before it can get nutrients from elsewhere. It's like packing a massive survival kit.
A self-contained starter pack for life.
Okay.
So these huge primary oocytes, they also undergo meiosis.
They do. 2 meiotic divisions, just like the primary spermatocytes. But how they divide is the crucial difference.
Unequal division, is that it?
Exactly. Unequal cytoplasmic division. When the primary oocyte divides in meiosis the first, almost all that precious cytoplasm, all those stored resources, goes to just one daughter cell, which becomes the secondary oocyte.
And the other cell.
The other cell is tiny. It's called the first polar body. It gets basically just a set of chromosomes and very little cytoplasm. It's essentially a way to discard half the chromosomes while keeping the resources concentrated.
And then the secondary oocyte divides again in meiosis the second.
Yes, and usually only if fertilization occurs. And again, the division is unequal. It produces the large functional egg cell, ovum, and another tiny polar body. The first polar body might also divide. So the bottom line is...
From one primary oocyte, you only get one functional egg.
Precisely. One large resource-loaded egg plus 2 or 3 small polar bodies that typically just degenerate. It conserves all those carefully accumulated materials for the potential embryo.
So connecting the dots. Spermatogenesis gives millions of tiny mobile cells built for speed and numbers. Oogenesis gives one or a few massive cells packed with resources for development. 2 totally different but really elegant strategies.
Perfectly put. Elegant solutions to the same biological challenge.
Okay, so we've got our specialized gametes. Now for the main event, fertilization. The moment they meet, and this raises a big question, right? How does the sperm arriving trigger the egg, which is relatively inactive, to suddenly burst into developmental activity?
Yeah, that activation was a puzzle for a long time. What's the actual molecular switch that gets flipped when the sperm makes contact? How does it turn on the egg's metabolic engine?
But there have been breakthroughs.
Oh, definitely. Some really cool work was done, for instance, with the sperm of Eurychus, a type of marine worm. Researchers Cross, Hornetto, and Goldsamaro, they managed to isolate a specific protein from the sperm's acrosome, that cap on the sperm head.
A protein. What was special about it?
It was a very basic protein, lots of lysine and arginine. And the amazing thing they found was that this protein, just on its own, could not only bind to Eurychus' eggs, but could actually activate them.
Just the protein itself, without the rest of the sperm.
Exactly. When they treated eggs with just this purified protein, the eggs started showing signs of activation. A surface coat lifted off, the nucleus inside started to break down, polar bodies formed. Basically things you see in normal fertilization.
Wow. So it mimicked fertilization.
It did. There was even a temporary electrical change in the egg membrane, a positive shift, similar to what happens right at fertilization. It strongly suggests that this kind of protein, or something very similar, might be the actual key, the specific molecule on the sperm that unlocks the egg's developmental program.
The molecular trigger, okay, so that gives us an idea of the what, but the how fast and how coordinated everything happens after that initial trigger.
Yeah.
That's another story, right? And sea urchins are key here.
Right. Sea urchin are fantastic models for this. You can get tons of eggs and sperm easily, mix them in seawater, and just watch the whole process unfold under the microscope. It lets you time everything.
And it happens quickly.
Unbelievably quickly. We're talking a cascade of major physiological and biochemical changes kicking off within seconds of the sperm contacting the egg. It's not just one thing, it's a whole programmed sequence.
Okay, seconds. Let's try and walk through this. What's first?
First, almost instantly, there's an influx of sodium ions into the egg.
Okay, sodium rushes in. Then
Then, calcium ions are released from storage depots inside the egg. This calcium flood is a huge internal signal.
A signal for what?
It triggers the cortical granule reaction. These little vesicles just under the egg surface fuse with the membrane and release their contents outside. This causes another big influx of sodium and helps lift off the fertilization envelope.
Is that the block to polyspermy, preventing more sperm from getting in?
That's a major part of it, yes, creating that physical barrier. Then, still within seconds or the first minute, you see metabolic shifts like NAD converting to NADP, a key cofactor change. Oxygen consumption shoots up.
The egg's engine is revving up.
Totally. The fertilization membrane finishes forming that complete barrier. The inside of the egg becomes less acidic. The intercellular pH rises.
Less acidic, okay. And then
Protein synthesis ramps up significantly. The egg starts making new proteins. Transport systems in the membrane get activated, ready to bring in materials.
wait, this is all still within the first few minutes. Electrical changes, calcium floods, barriers forming, metabolism kicking in, protein synthesis starting. It sounds incredibly intense.
It is. It's a highly orchestrated, rapid-fire activation. And it continues. Within several minutes, the sperm and egg nuclei, the pronuclei, migrate towards each other and fuse.
Combining the genetic material.
Right. That fusion triggers DNA synthesis, replicating the combined DNA. And then, relatively soon after, maybe within an hour or so, depending on the species and temperature, you get the first cleavage. The zygote divides into two cells. Development is officially underway.
That sequence, it really drives home that fertilization isn't just a simple merger. It's the initiation of a complex, pre-programmed cascade, turning that dormant egg into an actively developing zygote almost instantly.
It's a fertilization program, absolutely. Tightly coordinated and incredibly efficient.
And none of this can happen unless the right sperm finds the right egg. There's a recognition step first, isn't there?
Oh, absolutely critical. It's not random. There's species-specific recognition, often involving proteins on the sperm surface like bindin in sea urchins that act like a key.
A key that fits.
That fits specific sugar molecules like a lock on the egg's outer coat or membrane. Only the correct key, sperm protein, fits the correct lock, egg receptor for that species. It ensures you don't get cross-species fertilization inappropriately.
Nature's quality control again. So bringing it all together, fertilization is monumental. It's the fusion that starts a unique individual, combining parental genes.
And initiating that whole complex developmental journey. It's not just two cells meeting, it's the spark that ignites a lifetime of development.
It's been quite the journey for us today, too. From watching how sperm and eggs are meticulously crafted, those contrasting strategies of spermatogenesis and oogenesis,
Right, the quantity versus quality approach.
to witnessing that incredibly rapid, precise cascade of events during fertilization triggered by potentially a single protein. It's just layers upon layers of complexity.
It really is. Seeing how nature achieves this balance, how it activates development with such speed and precision, it's humbling, really. Cellular coordination at its finest, with consequences that shape all of life.
And just to leave everyone thinking, there's a question raised in the source material that really sticks with you. How can a tiny sperm, fusing with maybe only 0.00002% of the egg's surface, how can that tiny interaction trigger such a massive wave of changes throughout the entire egg, creating the new zygote?
That really puts it in perspective, doesn't it? Such a small initial touch setting off such a profound transformation. It highlights just how much intricate signaling and amplification must be going on inside that egg, and how much more there probably is still to understand about these very first moments of life. It definitely invites continued curiosity.